afternoon, I'm Caitlin here with Channel 10 News. Disaster has struck at a local supermarket where reports have alleged that there is only one bottle of hand sanitizer left. Our reporter Sarah is on the scene now. Sarah, do you have an update on the situation?
Good afternoon, Caitlin. You can see the chaos that has ensued behind me following the news that there is only one bottle of hand sanitizer left worldwide. There has been a disturbance between 9 supermarket goers, all after the final bottle of sanitizer. Local authorities have made their way to the scene and reportedly interviewed the members of the public. With assistance from local scientists, one member of the public will receive the final bottle of sanitizer. This is a worldwide crisis, and all people are urged to use the remains of their sanitation products in moderation until a solution is found.
Thank you, Sarah. We will now cross live to Dr. Strang, Director of the local Science Centre for Personal and Community Health, for an update on the situation.
Hello Caitlin. My team is working hard down here alongside the local authorities to interview the 9 members of the public. The interview process will allow us to shed some light on the severity of the situation. My team and I are in the process of conducting interviews and creating a criteria in order to make the fairest choice possible. This is what the 9 candidates have to say.
So what events leading up to now make you deserving of the final hand sanitizer bottle?
I'm a cleaner at the local supermarket and I've been cleaning with my hands all day.
I've been preparing lunches since 8 a.m. this morning.
I haven't washed my hands since 9 a.m. and my hands are filthy, especially with this baby.
At least you haven't been working in a bank all day. I've been handling everyone's money.
I'm not as young as I used to be and I always have to hold on to those filthy elevator handles.
I've heard mobile phones are the dirtiest objects around and I've been texting all day.
I've just been at the ATM.
I'm a professional runner and I'm always touching the bottom of my shoes. I've just been pushing around the shopping trolley for the last 20 minutes and between you and me, it's probably the dirtiest of them all.
As you can see, Caitlin, the following investigation will be challenging. However, with assistance from local experts, we will make sense of the public's needs and end this chaos.
For us here at the Science Centre for Personal and Community Health, it is of utmost importance that a fair and justified decision be made in regards to ensuring the highest quality hygiene services are provided to the individual most in need. It is for this reason that the focus of our inquiry will be on determining which surface contains the most bacteria and thus produces the most mould growth. In order to involve the community, a Year 6 class from a local primary school will assist in the investigation.
We pride ourselves on being up to date with the most current scientific information. So before we introduce the experiment, let's take a look at what mould actually is. Mould naturally exists in our everyday environment. It can grow on walls, bathroom tiles, carpet and food. Mould belongs to a group of microorganisms called fungi, but can be subcategorized as allergenic, pathogenic and toxigenic. These different types of mould have the potential to cause health problems such as respiratory illness, headaches or sinus problems. It is thus important for all community members, including children and adults alike, to understand the repercussions of exposure to a variety of bacterium. In order to determine who will receive the final bottle of sanitizer, the exposed inner surface of a potato will be swatched on the last surface touched by every member of the public. Our prediction is that the surfaces that are exposed to frequent public use will facilitate greater mould growth than those that are cleaned regularly in the home environment. With this in mind, the experiment was undertaken as follows.
5 potatoes were sliced into 10 equal portions. Each potato was wiped against its tested surface for approximately 10 seconds and immediately placed into a sealed Ziploc bag. important growth conditions for mould: temperature, humidity, and substrate. For this reason, each potato was then placed into the controlled physical environment of a 9 by 9 cm cardboard box. In order to maintain a controlled growth environment, the 10 boxes were then stored in a bathroom for a period of 7 days, as the conditions must exist simultaneously and consistently over a certain period of time to enable fungoid growth. As dampness, warmth, oxygen, and the absence of light affect mould growth, this location was considered the most favourable. Each day at a specified time, the mould growth was measured using a clear plastic grid and photographed to track changes. The mould growth of each potato was recorded on a spreadsheet out of a total of 32.
Good evening, I'm Caitlin here with Channel 10 News. Tonight we are here to present an exclusive update regarding the global sanitizer crisis. Our behind-the-scenes correspondents have just received word that scientists from the Science Centre for Personal and Community Health have concluded their investigation and finalised their decision. Sarah, we will cross live to you at the scene.
Thank you, Caitlin. The atmosphere here is indescribable. I am joined by our local expert who will present the final results. Welcome, Dr. Strang.
Good evening. It is with enormous anticipation that I am able to exclusively present the final results of our investigation. We understand the enormity of this decision, so we have cohesively and carefully collated the results in order to present the public with a justified and scientifically informed decision.
The results will initially be displayed from the least amount of mould produced to the greatest.
The graph currently pictured displays the results of a 7-day experiment whereby the percentage of mould growth on a potato sampled from a variety of surfaces was collected. As illustrated by the graph, the FPOS machine produced the greatest amount of mould compared to the public toilet and money producing the least amount. For this experiment, it was hypothesized that a surface that appears visibly contaminated would facilitate greater mould growth. When sampling the potatoes on the varied surfaces, the shopping trolley and the bottom of the shoe were perceived to contain the most bacteria. However, the hypothesis was disproved as the shopping trolley surface produced 43.75% of visible mould growth and the bottom of the shoe 56.25%. This can be compared to the mould of the FPOS machine covering 87.5% of the potato. Therefore, the investigation has drawn no correlation between the appearance of a surface and the growth of mould. For this experiment, it was predicted that a surface frequently exposed to public use would facilitate greater mould growth than those cleaned regularly. This was successfully proven for the FPOS machine, which is exposed to frequent public use but infrequent sanitization and cleaning. The surface with the second highest mould growth was hands, which are constantly exposed to a plethora of surfaces that are regularly and irregularly cleaned. However, other surfaces which were frequently exposed to public use contradicted this prediction, as they produce lower percentages of mould growth. These surfaces included the escalator handle, shopping trolley, money, and a public toilet. A possible source of error when collecting the data was sampling areas with inconsistent cleaning schedules. This may have influenced the results, as some surfaces would have been exposed to cleaning products at different times, rather than each surface having the same amount of time exposed to its surrounding environment. To illustrate this, the public toilet had been recently cleaned. Therefore, products such as bleach may have been used to sanitize the toilet and thus affected the viability of swatching the surface. Although the data obtained does not present any significant anomalies, rapid mould growth over the final 2 days were observed for the kitchen bench and hand surfaces. To explain this discrepancy, the 6 successive phases of mould growth can be applied. Initial growth lag phase, acceleration phase, log growth phase, delay phase, stationary phase, and declining growth phase.
In the first phase, initial growth lag phase, the bacteria's metabolism is activated. However, the main number of cells remain constant. This is evident on the days outlined on the graph for the FPOS machine, shopping trolley, and the bottom of the shoe. The duration of this phase depends on the temperature and the humidity at the spores. The acceleration phase then takes place, as shown on the graph for the shopping trolley, FPOS machine, and kitchen bench. Following this phase, the mould growth begins to confine itself to the reproduction of its vegetative units, known as the log growth phase. As depicted on the graph, this stage was reached on different days due to each sampled surface containing different substrates. When the reproduction rate falls due to unfavourable ambient conditions, like lack of nutrients, the delay phase is reached. Not all surfaces demonstrate this stage due to a delay in activating the mould's metabolism. Finally, the stationary phase takes place, whereby the number of germs remain constant as a result of new cells forming and others dying off. When the existing nutrients become more and more scarce and the toxic metabolism products are created, the fungus culture will begin to die off after the stationary phase. However, due to the investigation being carried out for 7 days, it was unable to be deciphered when and if the final stage would be reached. Further research shows that the properties of a surface are the decisive factors for initiating mycelium growth and mould formation. This infers that contaminations by dust, fingerprints, air pollution, or human perspirations are sufficient for the formation of a layer, even on sterile media. This may be a thin layer, but it enables spore germination and permits mycelium growth to take place, even though this is slightly delayed. This validates the mycelium growth on the controlled potato, which became evident from days 6 to 7. Although all potatoes were provided with optimal conditions to facilitate mould growth over a 7-day period, the varied results can be explained due to the diverse properties of each sampled surface. The swatch surfaces were contaminated by different amounts of dust, bacteria, and pollutants. This affects the fungus species, as the spores can be unicellular or polycellular, consist of different geometries, and occur in different quantities. Consequently, the successive stages of mould growth were reached at different times with different amounts of spore germination and mycelium growth.
These results imply that the growth and survival of living things are affected by physical conditions of their environment. Within a personal and community health context, there is a clear link to a primary school context, whereby students actively work to develop an understanding of both personal and community hygiene practices.
This investigation has drawn significant conclusions through enabling students to investigate the growth of mould spores on potatoes. These conclusions have assisted local authorities in deciphering which candidate is entitled to the final bottle of sanitizer. This investigation has shed considerable light on the greater issue of personal and community hygiene practices in our daily lives. With this knowledge, we have proven that the growth and survival of living things is affected by the physical conditions of their environment.
Thank you, Dr. Strang and team. Fortunately, normality will be restored back into the local community due to the rigorous data analysis and thorough research undertaken by the local experts. Although a conclusion has been drawn from this investigation, as scientists from the Personal and Community Health Centre have outlined, it does raise the concern of personal and community hygiene practices in our daily lives. We have to ask ourselves, is our community really as clean as we think? Back to you, Caitlin.
Thank you, Sarah. That's all we have time for tonight. From myself and the team, we wish you the cleanest of luck. Thank you and good night.